Is that a moon? A space station?
That's no moon. It's a nucleus.
Yeah, so in order to get into the nucleus, we gotta get through the nuclear membrane.
Not everything in the cell is good for the DNA, so the nuclear membrane plays an important role in protecting it and keeping it safe.
Well, hey there, officer. Just coming through on the official tour. Alright, you have a good day now. Oh man, I can't wait to see some DNA. I hope there's a replication fork happening in there. Howdy, we're hoping to see a replication fork today. Do you know where we could find one?
Excuse me, do I look like a tour guide? Do I look like a receptionist? I spent 6 years getting my PhD in nuclear logistics with a specialty in phasic transitions. I didn't go to the Robert Hooke College for the dividing arts to tell tourists about where the nearest replication fork is. There are 3.2 billion nucleotides in here, all woven into 23 unique chromosomes, each backed up with an extra copy. And all 3.2 billion of those nucleotides must be properly placed, sequenced, and duplicated every time we divide. You know whose job that is? It's mine. That's my job, and here you come.
Whoa, sorry. I was just curious. I'm sorry.
Oh, no, it's me. Of course I know. It's S phase, and things are complicated right now. The nearest replication fork is chromosome 19, 1.5 bases, and just go through the door. Have a good day.
Well, thanks. We appreciate your time. Good luck with S phase. Welcome to the nucleus. 3.2 billion nucleotides of pure U, all locked up right here in the cell. It's magnificent, isn't it?
Wow, it's got these cool springy things and blue lines.
You don't know what you're looking at, do you?
Yeah, I have a secret to tell you.
What? How could you not know what DNA is? You're a science teacher. We need to remedy this right now. Let's get you informed about this beautiful life-giving structure known simply as DNA, or scientifically, deoxyribonucleic acid. It's a double-stranded molecule that contains the code of life within its bonds. It's a fascinating structure that we're going to take a closer look at right now. DNA is just a long strand of nucleotides, which are repeating molecules made of three things. First are the bases. At the inside, we have T, A, C, and G, or also known as thymine, adenine, cytosine, and guanine. Now those terms are so long that we'll probably just stick to the first letter. Next, we have a phosphate backbone, along with deoxyribose, the sugar that connects everything together. So this is the simplified version, but let's run this through the deabstractifier and look at the molecular structure of these compounds. Alright, so now you can see there's two different groups of bases. We have purines, which are double ringed, and pyrimidines, which have a single ring. We throw on a deoxyribose and a phosphate group, and now we've got some nice looking nucleotides. You'll notice that A and T, which always pair together, have 2 hydrogen bonds, while C and G, which also always pair, have 3 hydrogen bonds.
So let me get this straight. DNA is just composed of repeating monomers called nucleotides, and each of these nucleotides has a phosphate group, a deoxyribose sugar, and a nucleobase, which could be one of 4, like cytosine, guanine, adenine, or thymine. Is that it?
Yeah, the important thing to remember is that these nucleotides are just one little part, and they get chained together to create the whole DNA molecule. time I told you about the central dogma of biology, a story that begins with DNA, the molecule of life, best called because it holds the blueprints for every single protein within our cell. Now we need to remember that proteins are the little machines that keep our cell running. So, in order to do this, DNA actually spins off a sister molecule known as RNA through a process called transcription. RNA then is free to leave the nucleus into the scary cytoplasm, where it will be read and turned into proteins through a process known as translation. Now this whole chain, DNA to RNA to protein, is known as the central dogma of biology. And it works pretty well until the cell needs to divide, at which point it needs to replicate its DNA so that the two new cells both have full copies of our DNA and all of the proteins that are required to keep the cell running. Now let's take a look at DNA and you. Within your DNA are specific sequences of nucleotides known as genes, and every gene codes for a protein, like Mr. Ribosome here. Every gene is also backed up by a second copy because you get one copy from both mom and dad. Now, the versions of those genes might be different. So one gene might come in many different forms, known as various alleles. And it's this combination of alleles which gives rise to our observable characteristics or phenotype. Let's take a look at eye color, for example. Depending on our combination of alleles, we might have blue or brown eyes. And so it goes with most of our characteristics, like hair color, or even something so simple as freckles. And this is why we consider DNA to be the building block of life, because it's part of that special sauce that makes you, you. Alright, I think that's all I have to say about DNA. We better get back to the cell. Mr. Ribosome's probably looking for us.
Well, hey there, it's Mr. Z from the future. That sure was an exciting episode. I learned all about the nucleus, which stores our DNA. It even has a membrane that's a protective barrier against bad things. Heck, I even learned about the structure of DNA again. It's made of nucleotides, which each have a phosphate group, a deoxyribose, and a nucleobase. And the sequence of those bases contain the information to make proteins and all of our unique characteristics. Oh, I can't wait for next week's episode. See you then.